So in this experiment, we tested the effects of different substrates on enzyme activity, different temperatures, as well as different pHs. During the first set of trials, we noticed that peroxide, of course, reacted the best with peroxidase. And the different substrates, such as alcohol and water, did not work as well. This goes back to the lock and key theory. So the enzyme was the same, our figurative lock. However, the key that we were taking to it was different each time. And only one of these substrates is going to fit into our enzyme peroxidase properly in order to initiate a reaction. Next one was our different sets of temperatures. So with our room temperature, the enzyme worked properly. This is roughly the temperature that the enzyme would normally be inside an organism. When we reduced the temperature of the enzyme and the substrate, we noticed that it wasn't active at first. And this gets back to our activation energy. So when it was very cold, there isn't enough activation energy to initiate this reaction. And then once we started to warm it up, all of a sudden, the reaction began to start. Now, when we ran that same trial with hot peroxide and hot potatoes, the potatoes did not react. And if we were to take these potatoes that have been heated up and allow them to cool to room temperature and take room temperature peroxide and put them together, we would still see no reaction. That is because the enzymes in these potatoes have become denatured. So that lock, that is the enzyme, has been smashed with a hammer, and now the key will not fit into it. When enzymes are denatured, we consider this a one-way process, and so cooling it down will not allow that protein to fold back into the correct shape in order to work again. So this is denatured and it will never work again. Lastly, we used glacial acetic acid to acidify a solution of hydrogen peroxide and compared that to hydrogen peroxide that is roughly neutral. And once again, the pH of our acidic solution was beginning to denature our protein. Proteins have a range of pH in which they can work. Once the solution that they are in is either too basic or too acidic, the efficiency of these enzymes starts to decrease drastically.